How do germ cell tumors originate in testicular cancer? Imagine the earliest moments of human development, when tiny cells are just beginning their journey to become the reproductive cells that will carry on our species. In males, these are called germ cells, which develop into sperm. Germ cell tumors in the testicles actually start from these very cells, but sometimes, things don't go as they should. To understand how these tumors form, we need to look back to fetal development and the biology of these special cells. During early pregnancy, primordial germ cells are the first to form. They migrate to the developing gonads, which will become the testicles in males. Once there, these cells are called gonocytes, and they are still immature. Normally, these germ cells follow a precise developmental path, maturing into sperm cells through a carefully regulated process. But in some cases, this process gets disrupted. Instead of maturing normally, some of these early germ cells stay in an immature and differentiated state. These abnormal cells can linger in the testicle for years without causing any symptoms, often remaining hidden. The main precursor to most testicular germ cell tumors is a condition called germ cell neoplasia in situ, or GCNS. This condition was once called carcinoma in situ. GCNS cells are basically arrested germ cells that look like embryonic cells. They still express certain stem cell markers, which means they retain some features of early development. These cells are thought to originate from the same early germ cells that failed to mature properly during fetal development. Usually, GCNS remains dormant during childhood and only becomes active after puberty, when hormonal changes stimulate these abnormal cells to start dividing. This is why testicular cancer is most common in young men, typically between the ages of 15 and 35. Genetics and environmental factors also play a role in the development of these tumors. Certain genetic conditions that affect testicular development increase the risk. Changes in molecular pathways, like those involving hormones, the KIT gene, or the DMRT1 gene, can influence whether these early germ cells continue to grow abnormally. Epigenetic changes, which are modifications that affect gene activity without changing the DNA sequence, may also contribute to this process. These changes can interfere with how germ cells mature, increasing the chance that GCNS will develop into invasive cancer. Testicular germ cell tumors are mainly classified into two types, seminomas and nonseminomas. Seminomas resemble the original primordial germ cells and tend to grow more slowly. Nonseminomas are more diverse. They can differentiate into various tissue types, reflecting their origin from pluripotent cells. Many tumors contain a mix of both types, which can influence treatment options and prognosis. Understanding how these tumors originate helps doctors develop better screening and treatment strategies. It also highlights how early life events and genetic factors can influence cancer risk later in life. For example, men with a history of undescended testicles or other developmental issues are at higher risk and should be monitored carefully. Advances in molecular testing and imaging are making it easier to detect these tumors early, improving outcomes. In summary, testicular germ cell tumors start from early germ cells that fail to mature properly during fetal development. These arrested cells can stay dormant for years, then become active after puberty, forming tumors. Both genetic predispositions and environmental influences contribute to this process, which involves complex molecular and epigenetic changes. Recognizing these origins helps distinguish testicular cancer from other reproductive cancers, like cervical cancer, which arise from different cell types. If you have concerns about testicular health, it's always best to consult a healthcare professional for personalized advice.